you for the opportunity to present our work here. So my name is Yifan, and I am a PhD student at the University of Waterloo. So today I'm going to present our work on modeling microbiome trait associations with taxonomy-adaptive neural networks. So microbiome research has two missions. The first is to predict disease status or other traits from microbiome profiles. And the other is to identify critical microbes associated with diseases. And these two missions are fundamental in understanding the role of microbiome in human health and diseases. And so it is very important to have an accurate modeling of microbiome trace associations. However, there are several challenges to do this. The first is potential inaccurate profiling may introduce uncertainties in read mappings, and this can introduce noisy features and hinder the downstream analysis. And the second, the curse of dimensionality is a common issue in microbiome data. And so we usually have far more features than the sample size. And this issue can lead to overfitting and make it very hard to train models effectively. And the third is the imbalanced dataset size. So we have many small datasets, but very few large datasets. And this imbalance complicates the analysis and impedes the applications of more powerful machine learning models like deep learning. So an ideal computational model should achieve a balance among three properties. The first is predictability, which refers to the model's ability to predict the disease status accurately from the microbiome profiles. And the second is interpretability, which refers to the model's ability to give an explanation for its prediction, which is essential for scientific discovery. And the third is trainability, which refers to the model's ability to learn effectively from limited data and generalize well beyond it. Achieving such a balance is very important for developing a robust and reliable model for microbiome disease associations. The question is, how can we achieve such a model? So we are facing a modeling dilemma. So each model may have its strengths or weakness in terms of these three properties. For example, linear model is interpretable and very suitable for small data, but may lack predictive power. Random forest has very strong predictive power and works quite well with high-dimensional, low sample size data, but it may be hard to explain. And the MLP, multi-layer perceptron, it can capture very complex relationships in the data, but it requires large data sets to train. It's also like a black box, which is really hard to explain its decision process. MIOSTONE, our proposed model, aims to achieve a balance among these three properties and address the three challenges which I mentioned before. So how are we doing this? So we leverage the taxonomic hierarchy, which is inherent correlation structure among taxa to mitigate this dilemma. So the taxonomy can provide very valuable information about the functional and evolutionary similarities between microbes, which can be informative for predicting disease status. And so we are trying to encode this taxonomic information directly into our architecture. So by aggregating the taxonomically related features, and so the milestone can capture the complex interactions among them and address the three challenges. So here are some details. So first, to counteract the potential profiling inaccuracies, so we employ data-driven taxonomic aggregation strategy. So this strategy uses the taxonomic hierarchy to aggregate information. So less informative features tend to be grouped together, and the more informative features persist. And so in this way, the impact of profiling inaccuracy will be minimized. The second, to address the curse of dimensionality, we employ a taxonomic adaptive pruning mechanism to prune the full connected neural network layers, which can effectively reduce the number of parameters and prevent overfitting. And the third, to handle small data sets, the milestone can apply a knowledge transfer approach, basically the transfer learning approach. So we can pre-train the models on a larger data set and then fine-tune on the smaller data set. And this is achievable because the milestone's taxonomic adaptive architecture, which basically facilitate the encoding of transferable knowledge from the related microbial features. And here are the experimental results. So we evaluate the milestone's predictive performance on several publicly available datasets. And one example is the inflammatory bowel disease dataset, the IBD dataset. It includes Crohn's disease and ulcerative colitis. And it has 174 samples with more than 5,000 taxa. And so this is very typical high dimensional low sample size data. And our task is to predict the subtype of the IBD. And we train all the models with 5-fold cross-validation with 20 random seeds. And we evaluate the performance with AUPRC and AUROC. So here's the results. We also perform a pair t-test to assess the statistical significance of the performance difference between milestone and other models. So we compared milestone with other machine learning models, including random forest, SVM, MLP, and other two deep learning-based techniques. And from this figure, we can see the milestone can consistently outperform other models, and which highlights its effect in modeling the associations. And the second is about the transfer learning. So milestone can improve predictive performance in sample limit tasks through knowledge transfer. So we validate this approach with datasets for the IBD disease. So it was pre-trained on a larger dataset called HMP2, and we evaluate with 3 different tasks on the IBD dataset: direct prediction on IBD, fine-tuning on IBD, and the training from scratch. And we also use AUPRC and AURC to as the metrics. Now here are the results. We can see fine-tuning can significantly improve the AURC and AUPRC scores compared to training from scratch. And Milestone also offers interpretable results for identifying microbial biomarkers. So here's a visualization of the importance score assigned to each genus. So we use the feature attribution method like deep lift or SHARP to compute the importance scores. And here's the visualization. And the features with high scores are highlighted, and they are supported by literature evidence. So basically the top 10 genus are highlighted, and we provided the PMID to support that. And here's one example, the Prevotella, which was found to be an indicator differentiating UC and CD. And in summary, so MIOSTONE serves as an effective predictive model, and it not only accurately predicts microbiome-trait associations, but also offers interpretability for scientific discovery. And here are the key ideas. So we reduce the feature noise by aggregating taxa in a data-driven way, and we mitigate curse of dimensionality by a taxonomy-adaptive architecture, and we use knowledge transfer to make it applicable to small data sets. And we believe it can facilitate in silico investigation into the biological mechanism underlying microbiome trait associations. And that concludes my presentation. Thank you.
Thank you. Any questions? Okay, please.
What type of neural network are you pre-training exactly?
We pre-train the MIOSTONE and other 3 deep learning based methods. I think you can see here. So the MLP, PopCCN, and taxon. PopCCN and taxon are methods previously proposed for microbiome data.
Gotcha. Thank you.
Any other questions? Yep.
I was wondering if you could use the kind of weights you're learning and stuff like that in a generative way and make kind of like null data or like sample data using some of the stuff that went into MIOSTONE.
I'm not sure I get your question. So you mean just directly extract the weights from the trained model and used for other like tasks?
Right, like it kind of use it like in order to like generate data or like null data or like fake data.
I think it's possible, but we have never tried that before.
Yeah.
Since the taxonomic the structure is like shared by any like microbiome data set, so I think it's possible, and I think it's a very interesting idea. Yeah.
Cool. Thanks.